Well, in this lecture, we'll be talking about the peripheral nervous system, that is, the autonomic nervous system and the somatic motor system. Your book presents the autonomic motor system first, and then the somatic motor system. I'm going to present it in the other order, that is, the somatic motor system first, because it's a simpler system than is the autonomic nervous system, and I think it'll be easier to understand the autonomic nervous system once we talk about the somatic motor system. So with that, let's move on to the somatic motor system. First of all, it is the system in which case where we have the efferent neurons going out to skeletal muscle. So it is controlling the soma, that is the body functions, and keep in mind again, soma refers to body. It has one neuron, excuse me, between the CNS and the effector organ. In this case, the effector organ is the motor, the motor neuron communicating them, but the effector organ is the skeletal muscle. So the effector organ is the skeletal muscle. And it is under voluntary control. That is, conscious intent makes skeletal muscle move because, of course, it activates the motor neurons going out to the skeletal muscle. Whereas when we talk about the autonomic nervous system, we'll see that that is not under voluntary control. So the anatomy of the of the somatic motor system. We've already studied synapses, and what we're going to be talking here is a special case of the synapse. First of all, the motor neurons. The cell body of the motor neuron resides within the ventral horn of the spinal cord, and it sends an axon out through the ventral root into spinal nerves, and then this, of course, synapses on muscle cells. The neurotransmitter that is released is acetylcholine. And we've already talked about acetylcholine as a neurotransmitter before. We'll be dealing with some of that same material again. Acetylcholine binds to receptors in the skeletal muscle that are what we call nicotinic receptors or nicotinic cholinergic. Remember, there are two types of cholinergic receptors, nicotinic and muscarinic. These are the nicotinic. The receptor requires two molecules of acetylcholine. It's shown here. It's a little bit small in this diagram, but of course it comes out of your textbook. You have these two molecules of acetylcholine that bind to this receptor. It is a directly linked receptor. That is, the receptor and ion channel are one and the same molecule. And the ion channel is what we call a mixed cation channel. A cation is a positively charged ion. And it's called mixed because it allows both sodium and potassium, two different cations, to flow through it. Sodium is, of course, more concentrated outside the cell than inside the cell. And potassium is more concentrated inside the cell than outside. So sodium moves in and potassium moves out. Now this causes depolarization of the muscle cell membrane. The reason it does is that you have a lot more sodium coming in than potassium going out. Therefore, there's more positive charge coming into the cell, and that, of course, is going to cause depolarization. This depolarization is, however, a graded potential. And as with any system where we're looking at graded potentials, if it's above threshold, it'll generate an action potential. It does this by, again, activating voltage-gated sodium channels, just like we see in neurons. So as acetylcholine is released, binds to the nicotinic receptor, which, again, there's more sodium coming in than potassium going out. This causes a depolarization, which is going to open these voltage-gated sodium channels, which, of course, is going to allow sodium to move in, generating an action potential. Now, we've talked about a motor neuron coming in and sending an axon out to and terminating on a muscle cell. But we need to deal with some of the terminology here. In this diagram, we have three different motor neurons sending their axons out to different populations of muscle cells. Each of these represents what we call a motor unit. A motor unit is a single motor neuron and all of the muscle fibers or muscle cells that it innervates. The term muscle fiber is the same as muscle cell. We use that term to refer to a muscle cell. So in this case, motor neuron 2 in red sends its axon out through here, and it terminates in 3 of these muscle cells or muscle fibers. This one, and this one. All of that is one motor unit, whereas the motor neuron 1, which is green, it's a dark green, but it sends its axon out, and it goes to 3 other motor fibers, actually 4 other. This one, and this one. So again, it's a motor neuron and all the muscle fibers that it innervates. Now I've referred to the communication between the motor neuron and the muscle cell as a synapse. And in fact, it is a synapse, but it has a special name. We refer to it as a neuromuscular junction, that it is a junction or a synapse between a neuron and a muscle cell. It, in fact, was the first synapse that was studied because it's in the peripheral nervous system. It's easier to get to than those in the brain, and it is enormous size-wise compared to the synapses in the central nervous system. The term terminal bouton is used to refer to the axon terminal because it was a French histologist that studied it, and it has a roughly button-like structure, and the word bouton is French for button. The postsynaptic membrane is referred to as the motor end plate. It is referred to that way because it is a specialized membrane that is different from the rest of the muscle cell. And for that matter, it's even different from most other synapses. It has this highly convoluted or invaginated surface, which produces an enormous increase in surface area, allowing far more cholinergic receptors as well as ion channels to be present and adjacent to the input from the terminal bouton. All motor neurons release acetylcholine, and the receptors on skeletal muscle cells are referred to as what we call nicotinic cholinergic receptors, because nicotine has greater affinity for this receptor than does acetylcholine itself. It's important you keep in mind that all neuromuscular junctions throughout the body are excitatory. So we always are going to have a depolarization occurring postsynaptically. Acetylcholinesterase is located here on the motor end plate, the postsynaptic surface, and it is there to degrade acetylcholine, which is going to limit the duration of the response. It would be difficult for us to have rapid and finely controlled muscle contractions if acetylcholine had a persistent effect. So by having the acetylcholinesterase here, it degrades acetylcholine fairly rapidly, helping to allow the muscle to relax so that it can contract again. So let's talk about the various steps that occur at the neuromuscular junction. First of all, the motor neuron coming in is what we call an alpha motor neuron, as we'll see when we get into subsequent lectures on muscle physiology, that there are other types of motor neuron, also delineated by Greek letters. But this is the alpha motor neuron coming in. And of course, whether or not it fires an action potential depends upon summation of EPSPs and IPSPs, just as we've already studied for other neurons. When the action potential travels down into this motor neuron, it's going to trigger the release of acetylcholine. So it's molecules of neurotransmitter and vesicles here are acetylcholine molecules. And these are going to produce what's called the end plate potential. This is, again, a graded potential. The end plate potential is a graded potential. It has a special name just because it was studied initially here at the motor end plate. But it is basically itself an EPSP of the membrane of the muscle cell. So now let's look at all of the steps that are involved. So we have the action potential moving down the alpha motor neuron. This, of course, as we've already studied about synaptic transmission, is going to activate voltage-gated calcium channels. As calcium comes into the membrane, it is going to cause the vesicles of neurotransmitter to move toward the membrane of the axon terminal, axon bouton, and fuse with it, and then allowing the neurotransmitter to be released into the synaptic space. So when acetylcholine binds to the nicotinic receptor here, it's going to, of course, cause opening of the ion channel, allowing sodium and potassium in. There's going to be a net movement of positive charge, causing depolarization. As I said before in the previous slide, we refer to this as the end plate potential. It's a special name that refers to an EPSP occurring here on the motor end plate. It is going to cause an action potential in the muscle cell. So as sodium comes in, it's causing depolarization of the membrane, and that depolarization is going to activate voltage-gated sodium channels. So we have the activation of these voltage-gated sodium channels, which is then going to produce an action potential, which is going to spread throughout the muscle, causing muscle contraction. Now, these steps we will not talk about in any detail until we get into a subsequent lecture on skeletal muscle physiology. But as that action potential spreads through the membrane, it's going to cause a variety of events, all of which are going to lead to muscle contraction. Now that pretty much finishes up the somatic motor system in terms of the synaptic structure and function. And as you can see, it's a pretty simple system. Now we're going to move to the autonomic nervous system, which is not nearly as simple. First of all, its primary function of the autonomic nervous system is to regulate organs, to maintain homeostasis. And as we saw in this diagram before, the efferent neurons coming into the somatic motor system, pretty simple, innervating skeletal muscle. Excuse me. The autonomic motor system is much more complicated. These afferent motor neurons are subdivided into two types, the sympathetic and the parasympathetic. It shows them being divided up again here into sympathetic and parasympathetic, but that's just the author's approach to separating the gastrointestinal system from all the other organs. But basically, you have these two halves of the autonomic nervous system, the sympathetic and parasympathetic, that project their axons out to virtually all visceral organs. And with that, we talk about a dual innervation. That is that all of these organs of the autonomic nervous system, there's a few exceptions, but by and large, all of them receive input from both the sympathetic and parasympathetic. So that both divisions innervate most of the effector organs. And they tend to oppose each other. The parasympathetic nervous system is what we call the rest and digest system. It is largely active when we are quiet and resting, as well as digesting meals, which of course is more typical of something we do while we're at rest. The sympathetic nervous system is quite often called the fight or flight response system. I dislike this terminology because it tends to suggest that it's only active when we're involved in motor combat, that when we're actually fighting with somebody or running away from somebody. But it is active whenever the body is not at rest. So just going for a moderate walk. It is certainly not something like fight or running, but it is still something that is not rest. It is distinctly different from rest. So whenever the body is trying to engage in some sort of pattern of activity other than rest, the sympathetic nervous system tends to be activated. Excuse me. Now, when we saw in the somatic motor system that there was actually one neuron, that we have a motor neuron coming out and going on to skeletal muscle cells. In the case of the autonomic nervous system, we have two neurons. We have what's called the preganglionic neuron here. I'm not sure exactly why my pen has changed. Here we go. The preganglionic neuron, which has a cell body that is within the central nervous system, it sends its axon out to what's called an autonomic ganglion. And that's why it's called preganglionic, because the axon exists prior to the ganglion itself. A ganglion is just a site where we have synapses out in the peripheral nervous system. The axon arising from the cell body here in the ganglion, because it exists after the ganglion, is called the postganglionic axon, and therefore the neuron itself is called postganglionic. Obviously, the term ganglionic would make a lot more sense because the cell body is here within the ganglion, but it was named because its axon exists after the ganglion. Excuse me. The ganglion is a communication between the preganglionic and postganglionic neuron, and it occurs in both the sympathetic and parasympathetic nervous system. We talk about the effector organs out here. We're talking about the organs that produce some sort of an effect in response to this input. So in that case, examples given here: cardiac muscle, that is the heart muscle; smooth muscle, whether or not it be vascular muscle, uterine muscle, gastrointestinal smooth muscle, urinary tract smooth muscle; glands, as well as adipose tissue, which we commonly call fat. So these are tissues and cells and organs that respond to input from the autonomic nervous system. We'll look at the sympathetic nervous system first. The preganglionic neurons originate in what's called the thoracolumbar spinal cord. Now, this diagram is a bit misleading. It looks like the cell body is coming from here in the ventral horn, but in fact, it's a little bit further back in an area that lies between the dorsal horn and the ventral horn, which is called the intermediolateral cell column, or also called the lateral horn. The preganglionic neurons, in most cases, come out and synapse in a series of ganglia called the sympathetic chain, which is just a series of ganglia that are fused. This occurs in most of the sympathetic nervous system. There are, however, a few so-called collateral ganglia that are located further away from the spinal cord. So we have a short preganglionic neuron and a long postganglionic neuron. Excuse me. There is a bit more complexity to this arrangement, and we'll talk about that here. In this case, the diagram is a little bit more obvious, that we have our preganglionic neuron with its cell body here in what's called the lateral horn. This little bit sticking out here is called the lateral horn. The cell body sends its axon down through the ventral root. Ventral root labeled here. This is the ventral root. Axon comes through in what's called the white ramus, which is a branch from the ventral root. It's called white because it looks white, has a lot of myelin in it. Synapses in the sympathetic chain. This structure here is a sympathetic chain. Synapses onto, I'll use another color, the postganglionic neuronal cell body here, which sends its axon out to the effector. So it's a little bit complicated in that we have this sort of looping structure here with a synapse involved in it. Parasympathetic nervous system, the preganglionic cell bodies are either in the brainstem or in the sacral spinal cord. Whereas the preganglionic neurons are short in the sympathetic nervous system. They are fairly long in the parasympathetic system, and correspondingly, the postganglionic neurons tend to be fairly short. So, whereas most of the sympathetic ganglia are fused in a chain that actually is located right close to the vertebral column, in the parasympathetic nervous system, the ganglia tend to be located fairly close to the effector organs themselves, and they are not fused. Now, it's easy, since we're talking about the autonomic nervous system, to consider it as a motor system exclusively. But in fact, it's what we call a mixed composition system. It has both efferent fibers as well as afferent fibers. So it has both motor neurons as well as sensory neurons in it. So the efferent fibers are the motor neurons we've been talking about, that is the pre- and post-ganglionic neurons for the sympathetic and parasympathetic nervous systems shown here. But in order for the autonomic nervous system to produce homeostasis, to regulate functions, you need to have some sort of afferent input to the central nervous system as a way of telling it the integrating systems up here. Remember, in homeostatic pathways, we always have integrating centers. You have to have sensory input coming into that integrating center to tell whether or not the efferent system is doing its job. So these afferent fibers transmit information from visceral receptors, that is, receptors located out in the organs themselves, into the central nervous system. Now, the example shown here is what we call the baroreceptor reflex, and I'm taking this material from a chapter that's later on in the book talking about the cardiovascular system. But I think it's an important system to jump into to understand how the autonomic nervous system works. In this case, we have these baroreceptors, baro meaning pressure. So these are blood pressure receptors. They are located in the carotid and artery and the aorta. They are located here at the carotid bifurcation, where the common carotid artery branches into the internal and external carotid artery, and there are also some of them located here in the aorta. The function of these is to make sure that there's enough blood pressure going up to the brain to maintain adequate blood flow to the brain. And they become activated when you get up quickly from either lying down or sitting position, because gravity tries to pull blood away from the brain. And the baroreceptors monitor this pressure and stop gravity from reducing blood pressure and making sure that there's enough blood flow going to the brain. So when there's a change in blood pressure, it's going to activate these neurons. If blood pressure goes down, if blood pressure goes up, of course, it will not activate them. So we have our afferent fibers coming from the carotid and aortic baroreceptors going up into our integrating center. Labeled here is the medullary cardiovascular control center because it's specific for cardiovascular control, and it's in the medulla oblongata. Excuse me. And this sends output to either the parasympathetic neurons or sympathetic neurons. Now, we haven't studied what these systems do yet, but I'll jump the gun just a little bit here. Parasympathetic outflow to the heart slows it, whereas the sympathetic outflow to the heart speeds it up. So if when you stand suddenly, blood pressure goes down, this signals these afferents telling the nervous system that you need to increase blood pressure. Well, you do that by increasing heart rate. So the sympathetic outflow to the heart, specifically the SA node, increases. Increasing heart rate, sympathetic outflow to the ventricles causes the heart to beat more forcefully, ejecting more blood. Going out to the arterial side of the system, causing these vessels to constrict so that more pressure is built up to drive more blood up to the brain, and causes the veins to constrict as a way of increasing the amount of blood coming back to the heart. Meanwhile, if we're causing all of this increase of activity of the sympathetic nervous system, remember, the systems oppose each other. So in that case, we would decrease the parasympathetic outflow to the heart, whose primary function is to decrease heart rate. So because we're trying to increase heart rate, we of course want to inhibit this effect that's trying to decrease heart rate. And again, the whole point here is that you can only do this if you have autonomic nerves that contain both efferent and afferent nerve fibers. You need to complete the loop, basically. So now let's take a look at the autonomic neurotransmitters and the receptors. On the top of our diagram here, we have the sympathetic nervous system. So right now, we're just going to be looking at the upper half of the diagram. We have three different arrangements. The predominant one is shown in this upper panel, where we have the cholinergic preganglionic neuron. We've already talked about the concept of preganglionic neuron. Again, preganglionic neuron releases acetylcholine. Acetylcholine binds to a nicotinic receptor, just like we saw in skeletal muscle. So it's the same receptor, mixed cation channel as well. The postganglionic cell body releases norepinephrine. So we call it an adrenergic neuron. Norepinephrine binds to adrenergic receptors. Now, there are 5 subtypes. We'll be talking a bit more about those in another slide or two. This is what's happening in most organs in the body, is what we have in this upper panel in terms of sympathetic input to most organs of the body. The adrenal gland has an adrenal medulla, which is at the center of it, an adrenal cortex. We're talking about the adrenal medulla in this particular case. Again, we have a cholinergic preganglionic neuron that stays the same. There is no ganglion here. The adrenal gland itself, specifically the adrenal medulla, embryologically started out as a ganglion. But rather than the cell bodies becoming postganglionic neurons, they became endocrine cells. And they synthesize primarily epinephrine. Actually, 80% of the hormone released is epinephrine, 20% is norepinephrine. Excuse me. So these neurons, which have developed into endocrine cells, release epinephrine, which binds again to adrenergic receptors. And again, there are 5 subtypes. We'll talk about them later. There is one special case in terms of neurotransmitter for the sympathetic nervous system. Again, keep in mind we're talking about the sympathetic nervous system here. And this is shown here. This is a deviation from the diagram in your book. I've labeled some things here. This, first of all, rather than just saying effector gland, is a sweat gland. But the important thing here for the control of sweat, again, we have a cholinergic preganglionic, of course, releases acetylcholine, binds to nicotinic receptors. And I forgot to mention up here in the adrenal gland, these are also nicotinic receptors. But the postganglionic neuron is not adrenergic. In terms of the control of sweat glands, it is cholinergic. So it's a special case. It releases acetylcholine, which binds to, in this case, muscarinic receptors, a different subtype of cholinergic receptor. I've mentioned it in a previous slide, or a previous lecture. It's named because the ligand with the greatest affinity for this receptor is muscarine, which comes from the mushroom Amanita muscaria. So, sweat glands are the special case of the sympathetic nervous system. And as you'll see, they actually look a lot more like a parasympathetic system. And we'll talk about that now. The parasympathetic system, you have your long cholinergic preganglionic neuron, again, releasing acetylcholine, binding to nicotinic receptors. So one thing to keep in mind here, nicotinic receptors are involved everywhere here. So they're not chemically distinguishing between the different systems. So we have our nicotinic receptors activating a cholinergic postganglionic neuron. So it's releasing acetylcholine, and in this case, again, activating muscarinic receptors, not nicotinic receptors. So this arrangement for the parasympathetic system is the same for all organs that the parasympathetic nervous system communicates with. And as you see, it parallels what we see in terms of the control of sweat glands. It's just that these preganglionic neurons that are controlling sweat are coming from the thoracic and lumbar spinal cord, which is, of course, where all of the other preganglionics from the sympathetic nervous system are coming from. These do not come from the brainstem and sacral spinal cord, whereas these preganglionic neurons do. So chemically, control of sweat looks like it's a parasympathetic system, but functionally it's a sympathetic system. So I mentioned 2 different types of cholinergic receptors in the previous slide, and again in the previous lecture. Let's talk a little bit more about them. The nicotinic receptors or muscarinic receptors. The nicotinic receptors, shown in this upper panel, again are requiring 2 molecules of acetylcholine to bind. It is the mixed cation channel. So sodium can flow in and potassium can flow out. Again, we have the net result is depolarization because more sodium is coming in than potassium leaving. And these nicotinic cholinergic receptors exist in both the sympathetic and parasympathetic nervous system. I made that point in the previous slide. They also exist in skeletal muscle. These receptors are the same receptors in terms of how they function. in autonomic ganglia as well as in skeletal muscle. So pharmacologically, there's no way to separate them, which means a drug that affects autonomic ganglia will affect skeletal muscle and vice versa. Muscarinic receptors are G protein coupled. The effect that they produce depends upon the target cell. That is, it depends upon what type of a signaling arrangement exists in the target cell. But both are possible that are shown in this diagram, and both do occur in some particular cell types. So we have here molecule of acetylcholine binding to a muscarinic receptor, which is coupled to a G protein. The G proteins can do one of two things. They can work on an ion channel, in which case we have our indirectly gated and slow-acting ion channel, which we've talked about before, or the G protein can work on an amplifier enzyme producing a second messenger. Excuse me. As I mentioned a couple slides ago, there are 5 subtypes of adrenergic receptors. There are the alpha 1, alpha 2, beta 1, beta 2, and beta 3. We only show alpha 1 and alpha 2 here, and then beta thrown together, and I'll explain why we're doing that. The alpha 1 receptor activates the phospholipase C system. It does this through the G type of G protein. So we have, in this case, norepinephrine or epinephrine, which are both ligands for the alpha 1 receptor binding to the alpha 1 adrenergic receptor, activating the G protein. And again, GS stimulates the amplifier enzyme. Don't get confused with the idea that GS has to stimulate adenylate cyclase. GS can stimulate whatever amplifier enzyme is involved. In this case, it's phospholipase C, which of course phospholipase C causes the production of IP3 and diacylglycerol. IP3 then works, of course, on the endoplasmic reticulum, causing calcium release. DAG activates protein kinase C. The alpha 2 receptor and all 3 of the beta adrenergic receptors, so beta 1, 2, and 3 here, all work this way. The alpha 2 receptor stimulates a G protein coupled receptor, but this is GI. GI inhibits the amplifier enzyme, which in this case is adenylate cyclase. So when either epinephrine or norepinephrine bind to the alpha 2 receptor, it inhibits adenylate cyclase. So what happens is that you do not get production of cyclic AMP. You do not get the activation of protein kinase A. You actually decrease this activity. Whereas the beta receptor increases this activity. It's acting through GS, which is a stimulatory protein. Now, one of the things you need to keep in mind is when we're talking about G protein so far, I've talked about how the beta and gamma subunits dissociate from the alpha subunit. The alpha subunit is the one that dictates whether or not that G protein is a GI or a GS, or there's actually several other designations as well. So it is the alpha subunit that gives it the inhibitory effect, and the alpha subunit that gives this the stimulatory effect. What you can also see from this is that alpha 2 receptors have the opposite effect of beta receptors. Alpha 2 receptors inhibit adenylate cyclase. Beta receptors activate adenylate cyclase. So that this is a way of causing very, very careful regulation of the activity of this adenylate cyclase. And we'll see some of the implications of that once we get into the autonomic nervous system and talk about some of the effects that these receptors produce. So now I want to talk about some of the details of the properties of these adrenergic receptors. Some of this material is in your book, but it's spread around, so I've put it all in one place. We have our alpha receptors here, signaling through G protein. The alpha 1 receptor works, remember, through the phospholipase C system, that is through IP3, adenosine triphosphate. Inositol triphosphate, and DAG or diacylglycerol. The alpha 2 receptor works through GI, and decreases cyclic AMP. Alpha receptors are located in the effector organs of the sympathetic nervous system. This is important. Alpha receptors are more common than beta adrenergic system. If you take a look throughout the body, there are far more alpha receptors than there are beta receptors. They are usually excitatory. So most of the effects that we attribute to alpha is actually alpha-1, because alpha-1 causes excitatory responses. Generally, in most organs, if you inhibit cyclic AMP, you actually decrease the function of that organ. And the affinities, that is, the ligands, norepinephrine and epinephrine, have, of course, affinity for the receptor as in any receptor. And for both alpha-1 and alpha-2, norepinephrine has a greater or equal affinity than epinephrine. Now, this is what separates alpha receptors from beta receptors. For 2 of the 3 beta receptors, epinephrine has greater affinity or equal affinity. So generally speaking, if it's an alpha receptor, norepinephrine has greater affinity. If it's a beta receptor, the safest thing to keep in mind is that it's probably that epinephrine has greater affinity for it. So let's look at the signaling now in the beta receptors. Again, they're G protein linked, but they're linked to GS, which increases cyclic AMP. By and large, beta receptors located in various places. Beta 1 in the heart, not exclusively, but that's where most of them are. And they increase, doesn't show up very well. For some reason, the arrow is not showing up very well here, but it increases heart rate and contractility. Beta 2 receptors generally cause relaxation of smooth muscle. So beta 2 receptors relax smooth muscle of the airways, making it easier to breathe. They relax the uterus, so they retard the birthing process. And they cause dilation of blood vessels going through skeletal muscle. And we'll talk about the significance of this once we get into the autonomic nervous system. Once we get into the cardiovascular nervous system. Beta 3 receptors are restricted to lipids or fat, so our fat deposits. And at this point, it's not really well understood how they work in the human body. I've already discussed the affinities of the beta receptors and contrasted them to the alpha receptors. Now, in the somatic system, we talked about the neuromuscular junction. In the autonomic nervous system, we talk about the neuroeffector junction, because the end organ, the effector organ, is receiving input from the motor neurons. Now, in most cases, the effector organ is smooth muscle. But, of course, the heart is composed of cardiac muscle, not smooth muscle. So that's one reason why we have this more general term, because the effectors are different types of muscle tissue in most cases. And some of them are glands. So first of all, it's the junction or synapse between the postganglionic neuron and the effector organ. And the neurotransmitter is stored in axon swellings that we call varicosities. Think about varicose veins, if you're familiar with them. So these are these swellings, sort of like a little string of pearls sort of arrangement along the axon, what's called the terminal axon here. What this does is it allows a single motor neuron coming in to send outflow to large areas of a single organ or a single tissue, and cause release of neurotransmitter at multiple locations all at one time. So it's a way of causing very widespread activation of the tissue or of the organ. So that we have, again, in this case, norepinephrine that's being released, but again by voltage-gated calcium channels that are being activated by the action potential coming down through the axon here, activating voltage-gated sodium and potassium channels just like we see in neurons. So, of course, there's more sodium coming in than there is potassium going out, causing the action potential to come in, activating the voltage-gated calcium channels, causing the neurotransmitter to be released. The important thing here, again, with these varicosities is that it's very widespread, very diffuse release of neurotransmitter throughout an entire organ. And just as we saw in other systems, in terms of the synapses, we need to look at the events that are occurring. The action potential arrives at the varicosity, activates voltage-gated calcium channels. This triggers excitosis of the neurotransmitter, and this, of course, is norepinephrine. It binds to the receptors on the effector organs, causes a response in the effector organ, and then the neurotransmitter is degraded or diffuses away or is taken up by reuptake pumps. A little bit of detail with these varicosities in terms of the handling of neurotransmitter. Again, we have our voltage-gated calcium channels. We have our, our vesicles, in this case for acetylcholine release from a postganglionic neuron for the parasympathetic system, and in this case, norepinephrine release from the postganglionic of the sympathetic nervous system. So in both cases, we have calcium coming in. We have vesicles of neurotransmitter. Those vesicles release the neurotransmitter. The neurotransmitter diffuses across the synaptic space, binding to receptors. The real difference is then what happens afterwards. For acetylcholine, we have the enzyme acetylcholinesterase that breaks down the acetylcholine into choline and acetate. The acetate is taken up by diffusion or by other tissue surrounding it, but the choline is pumped back up by a reuptake pump and reused in the synthesis of new acetylcholine. Norepinephrine, once it's released, there is no enzyme here, the way we saw here. There is no enzyme here. That norepinephrine either diffuses away or is taken up by a reuptake pump and again reused. The metabolism of norepinephrine is largely occurring with the mitochondria and the release of monoamine oxidase. And monoamine oxidase is an enzyme that can degrade norepinephrine. So how do we regulate autonomic function? Well, again, you have the dual innervation. So you have sympathetic system and the parasympathetic system. Sympathetic system generally is going to activate, let's say we're talking about heart, increase heart rate. Parasympathetic nervous system does the opposite. It decreases heart rate. And again, this is due to this dual innervation, that you have both branches of the autonomic nervous system coming into an organ, and remember, they oppose each other, which is why the parasympathetic system is largely active in rest, and the sympathetic is largely active whenever there's any sort of excitation that is a deviation from a restful state. And they, as I said, oppose each other. So if there's an increase in parasympathetic activity, it generally is coupled with a decrease in sympathetic activity, and vice versa. If you think about the idea of homeostasis and negative feedback, this is a perfect example that one system opposes the other. And with the two systems working together in this dual innervation, you actually have these continued oppositional forces that are keeping a very delicate balance in terms of visceral function. So again, we're going to go back to the baroreceptor reflex. We've seen the diagram here on the left side. Let's take a look at it in terms of a flow chart. So a person stands up. Whether or not they're lying down or sitting up, they stand up. The force of gravity causes blood to pool down in the feet. So blood is moving down toward the feet. This means, of course, that there is less blood flow going to the brain. Less blood pressure here. And, of course, we want to maintain adequate blood flow to the brain. So blood pressure is going to go down. It goes down very, very briefly. This is going to activate the baroreceptors. They will detect this decrease in blood pressure. Remember the concept of the error signal we talked about in homeostatic pathways? So this is going to decrease the action potentials of these neurons. Baroreceptors actually, their action potential correlates directly with blood pressure. So greater blood pressure means greater frequency of action potentials. So this is going to cause if blood pressure goes down, this is going to cause the frequency of action potentials in the baroreceptors to go down. They send their input up to the integrative center, in this case the vasomotor or cardiovascular control center, where integration occurs. This is going to cause a decrease in parasympathetic activity, because parasympathetic activity, again, would tend to slow the heart, and increase sympathetic activity, which is going to speed up the heart, make the heart beat more forcefully, and also increase the amount of constriction of blood vessels, which is going to help to increase blood pressure. This, as we say here, increasing blood pressure. And of course, this is a perfect example of negative feedback. Started out with a decrease of blood pressure. We now have an increase of blood pressure, restoring blood pressure to where it should have been. So let's talk about the systems and when they're active. Parasympathetic system is generally most active during quiet, relaxed states. It's what we call the rest and digest system, primarily because it increases gastrointestinal activity, so it's helping us to digest and absorb food, and decreases heart rate and blood pressure, which is largely consistent with a restful state. Sympathetic nervous system we refer to as the fight or flight response. It's basically, as I said, not so much restricted to mortal combat, but it's for emergency, stressful, and exercise situations. So whenever the body is removed from a restful state, the sympathetic nervous system becomes active. It increases heart rate and blood pressure, mobilizes energy stores. In order for other body tissues, or for that matter, even for the heart to become more active, you're going to need to have more energy available. Causes dilation of our pupils, and decreases gastrointestinal and urinary function. So this is a perfect example of how the two of them oppose each other, as you see right here. Besides on heart rate, they also oppose each other in terms of gastrointestinal function. This is one reason why it's not a good idea to eat too soon before exercise, because exercise, of course, activates the sympathetic nervous system, which is going to decrease gastrointestinal activity, which tends to result in cramping if you've just eaten a meal. Physiologists quite often talk about tonic activity. And what we mean by tonic activity, or sometimes we use the word tone, is the amount of moment-to-moment ongoing activity. The autonomic nervous system has tonic activity at rest. Even the sympathetic nervous system, which may be inhibited, still has a certain level of activity that may be lower than during exercise, but there's still a certain amount of ongoing activity. So both branches are active nearly all of the time. Example here that we have tonic sympathetic input to blood vessels. So we have a certain amount of activity here. This is intended to represent frequency of action potentials of the postganglionic neuron going to the blood vessel. And the vessel is either going to constrict with a greater number of action potentials, if you compare this here, or with fewer action potentials, it's going to dilate. So that it's almost as though the body is working like a driver with its foot on the gas and the brake at the same time, and it depends upon how much excitation or inhibition of the system, giving you some sort of intermediate ground in terms of what the ultimate level of activity is. For the parasympathetic nervous system, because it cannot affect blood vessels, it can only affect the heart, we show how it affects heart rate. So in this case, we have the heart receiving both sympathetic and parasympathetic input. There's a certain amount of ongoing activity, and during rest, we have a lower level of activity of the sympathetic neurons, but a greater level of activity of the parasympathetic neurons, basically keeping the heart rate slow. During exercise, there's greater activity of the sympathetic input, less activity in the parasympathetic input, but heart rate goes up because we have this greater level of activity from the sympathetics. But there is still a certain amount of tonic activity in each of these two halves, in both the parasympathetic and sympathetic. It's just in this case, we have more in the sympathetic and less in the parasympathetic, whereas in this case, we have more in the parasympathetic and less in the sympathetic itself. So we're talking about both their competition with each other, as well as the fact that they tend to have some level of tonic activity. So now let's take a look at tables talking about the overall effects. The parasympathetic nervous system versus the sympathetic nervous system. Parasympathetic nervous system decreases heart rate by working on what's called the SA node or the sinoatrial node. We'll be talking about this in more detail when we get into the cardiac system. But I assume by this point that you've had enough anatomy as your background that you've heard of the SA node, and it's best not to just hold off on all of this material until we get into the cardiac chapter. So basically, the parasympathetic nervous system decreases heart rate, decreases the conduction velocity, that is, the speed with which action potentials move through the AV node, moving down through the electrical conduction system into the ventricles. And your book says that it decreases the force of contraction of the ventricle. It says it's a small effect. It is so small that most books consider it non-existent. And the parasympathetic system has no effect on any of the blood vessels. So the thing to keep in mind, parasympathetic nervous system decreases heart rate primarily. Sympathetic nervous system increases heart rate through beta-1 receptors, increases conduction velocity, again, that is the movement of depolarization through the electrical conduction system of the heart, again by beta-1 receptors, and increases the force of contraction, again by beta-1 receptors. Sympathetic nervous system does work on blood vessels. It works on them in many ways. First of all, norepinephrine binding to alpha-1 receptors causes vasoconstriction, that is a decrease in diameter of the blood vessels. It lists vasoconstriction twice here. This should actually be venoconstriction, which is constriction of veins. So vasoconstriction is constriction of arteries. Venoconstriction is constriction of veins, which is also mediated by alpha-1 receptors. Now, epinephrine, when it's released, binding to beta-2 receptors, causes vasodilation. Now, this obviously opposes the vasoconstriction that's produced by alpha-1 receptors. When we get into the cardiovascular system, again, we'll talk about this in more detail. But at this point, what you need to be familiar with is the idea that norepinephrine is going to be released by neurons very rapidly during the onset of exercise. And of course, neurotransmitter works almost instantaneously. So sympathetic activation initially is going to be working through norepinephrine binding to alpha-1 receptors. Then let's say if you're going for a run, after several seconds or perhaps even a little longer, but certainly within a minute, there's going to be, because of greater level of activity, greater metabolic demand, there's going to be greater level of activation of the sympathetic nervous system. So you're going to start having epinephrine released from the adrenal gland. Epinephrine has a greater affinity for beta-2 receptors. So it is going to start causing vasodilation. Beta-2 receptors are largely restricted to those blood vessels that are going to skeletal muscle, whereas the alpha-1 receptors tend to be located in other vascular beds. So you're going to have vasoconstriction in other tissues and vasodilation in the blood vessels that are nourishing skeletal muscle, which makes sense. You want greater blood flow going to the skeletal muscle during exercise. And epinephrine also can cause vasodilation as well through binding to beta-2 receptors, but this is only likely to occur when you have very massive levels of sympathetic nervous system activity that is able to activate these receptors as well. Excuse me. Again, parasympathetic and sympathetic nervous system, they tend to oppose each other, whereas the airways contract because of parasympathetic input, that is bronchial muscle is airways. They relax when you activate the sympathetic nervous system. And again, it's beta-2 receptors working on smooth muscle. This, again, makes sense. If you think about exercise, when you're exercising, you want your airways to dilate so you can move air in and out more easily. And again, beta-2 receptors located on smooth muscle cause it to relax. And again, looking at this chart, most of the responses that are produced by the parasympathetic nervous system are the exact opposite of those produced by the sympathetic nervous system. As well as in the urinary system, reproductive tracts, we still have these opposing effects. In the skin as well, control of sweat glands. First of all, there is no parasympathetic input to sweat glands. You do have stimulation of sweat. I'm sorry, I misspoke here. Sweat glands. It says in your book that the parasympathetic nervous system stimulates secretion. I think that that's a mistake because it is largely through the effect of acetylcholine being released by the sympathetic nervous system. But again, the overall point on these four charts is that effects produced by the parasympathetic nervous system are, in most cases, the opposite, except, of course, where there are non-existent, of the responses produced by the sympathetic nervous system.